Hello, everyone, and welcome to our program, SERC Science to Go, Fun with Fungus. My name is Joanna, and I am a Programming and Outreach Coordinator with the Anne Arundel County Public Library, and I will be your host today. Programs like this are brought to you with support from the Anne Arundel County Public Library Foundation. To support our programs and all of the work we do, please consider honoring Anne Arundel County Public Library's 100th anniversary by donating $100 for 100 years. You can learn more at www.aacpl.net/100years. We also want to take a moment to thank the Brick Company and Smithsonian American Women's History Initiative for supporting and funding this program. For the best viewing experience, you can download the Zoom app to your computer or mobile device. We also have sign language interpretation and closed captions available. Throughout the program, please feel free to share any questions you may have for our presenters in the Q&A box. We'll have some time at the end to answer questions. And now, without further ado, I'd like to introduce Karen Mcdonald.
Hey everybody, so I'm the STEM program coordinator here at the Smithsonian Environmental Research Center, and I'm excited to bring you some really neat science today with Adventures with Fungus. So we're going to take our questions to the question and answer. So as we go along, if you need help with the activity or you have questions about some of the things the scientists ask, put that into the question and answer, and we'll be sure to try to get to those. So here's my question for you. Do you know what fungus is? See if you do. Type that in the chat. What's your best guess? Have you ever seen it on things in your refrigerator? Like on a piece of bread or even some old fruit maybe? So type that in the chat. Let us know what you think fungus is. So the theme for today's talk is fungus is your friend, because fungus is found in our forests and our food. And today we're going to learn more about that and about what our scientists do. Now remember, we love to see everybody working really hard. So if you want to post pictures of yourselves doing our activity or before or after your experiment, then you can use the hashtag #ScienceToGo, and you can post that on Twitter, Instagram, or Facebook. We'd love to see you and what you're doing with your experiments. So before we get to our scientists, we need to do a few things with our experiment to get everything ready and running while they're talking. So the first thing we want to do is push up our sleeves and get ready to do a little bit of science. All right, so go ahead and get your science kits ready. Open those up, and we're going to look at some of the supplies that we have inside. You should have a bottle, a balloon, some yeast and sugar. You're going to have a little baggie with some fungus and tweezers in there, and you're going to have a napkin and a notebook card. So get those ready. Now remember, wherever you're working, make sure that you don't get water or anything on your laptop or electronics, all right? So make sure that when you're setting up for this, you're not going to get anything wet, all right? Now, there are several experiments in the Science to Go booklet, which we had in your packets. If you get behind today, or if something doesn't make sense, you can go back to that packet. We're also going to record this talk, so if you want to go back and review it, or if you want to watch it again, that's going to be on our website live, but it'll be a little bit later, all right? Like I said, though, if you have questions, go ahead and put that into the question and answers, all right? So the first thing we're going to do to get ready is we need to start with our hydrating fungus. So to do that, I need you to get your little baggie out that has your tweezers. They're also called forceps. It has a little piece of sticky stuff in it, and it's got some dried mushroom that we put in there for you. Now, when you do that, you're going to empty that out into your hands. And when you empty that out into your hands, you should take your fungus and your mushroom and set those aside. Now, the little sticky thing in there, that's just tacky putty. So you want to take that and try to pull it out. There's going to be a little tiny glass lens, or actually I think it's plastic, that's going to be with it. Now, if you have trouble getting that putty out of your bag, you might need to turn it inside out to be able to get it out of your bag. But you want to take that putty and the little lens, your tweezers, and your fungus out. Now this is the part where we're going to need you to fill the baggie with a little bit of water. You're also going to need some water for your little bottle. And so what I want you to do is to get some warm water. It can just be tap water, just room temperature. And you're going to fill your bottle up you're going to put 2 fingers on the edge of your bottle, just like that. It can be adult fingers if you're a little kid, 3 fingers. And you're going to fill your bottle up. Add a little bit of extra because we want to fill this baggie just enough to cover your mushrooms so you can pour it into your bag. So go ahead and take this moment. I'll take a little pause here and fill up your bottle with warm water from the tap. And this is why it's important that you don't work around your electronics or computer. All right. And I'm going to do that real quick. I've got my little beaker here. You're going to use your sink or whatever you've got. And I'm going to pour some extra in there. All right, how are we doing? I know it may take just a minute for everyone to get to the sink and to get back. That's okay. All right, when you've got your water and when you're ready, you're going to take your little baggie and you're going to pour some water in, just enough to cover the bottom, and you're going to drop your mushroom down in there and make sure that you can cover your little mushroom bits up as best you can. Now, you can do this with any dehydrated mushroom you get at the store, or you could do the activity we're going to do with a mushroom from the grocery store that isn't even dried up. But for today, we gave you dried up ones because it's easier for us to send to you for this activity. Now, when you're done covering your mushroom up, you want to go ahead and seal up the top of the bag, make sure it's all the way sealed, and then just set that aside. You don't need that any longer, all right? And essentially what we're trying to do is we're just trying to get that dried up mushroom to inflate, so when we do our mushroom gill dissection, you'll have something to work with. All right, so go ahead and set that aside. Okay. All right, and you can use any cup or bowl to fill the bottle. Don't worry about having just specifically using this bottle. You can use anything to fill that baggie. It can be anything that you want to use. Now, in your bottle, you should have some water left over, or you should have some warm water in there. All right. Now, this water is what we're going to use with our yeast. Now, our yeast is this packet right here that we want to get going for our experiment. And we're going to talk about that in just a little bit. But what I want you to do is I want you to go ahead and open your yeast packet. All right, open that up. You might need scissors or you might need an adult to help you open it. All right, and you're going to very carefully pour the yeast into the water. Very carefully. It's okay if a little bit spills. Sometimes it's kind of hard not to. This is the same yeast that you might use for bread or for making food at home, like pizza dough. All right. Now, we want to also include with our yeast the sugar. All right. We need to feed our yeast. So we're going to take that sugar packet. We're going to open it up. And we're going to pour that in. It's a little tricky. It's okay if some spills. Okay, I'm going to pause here for just a minute and let everybody kind of catch up. So just to review what we just did. We had water in our bottle. We have our yeast that we poured in, and we poured in our sugar. All right. Now, if you want to, you can swirl it around just a little bit. Don't be too vigorous, but you can swirl it around. And yes, you do want to put all of the yeast in. You want to put everything in there, all right? So you can swirl it around, mix it up just a little bit. And the next thing we're going to need is the balloon. So make sure you get that ready, all right? Okay, so with the balloon, you're going to take this. It's really helpful to stretch it out because it's going to end up getting filled with gas. So you're going to need to take your balloon and stretch it a little bit. I put a smiley face on mine just because I think it's funny. But stretch your balloon a little bit. And if you're still working on pouring your yeast and sugar, that's okay. All right, so stretching that balloon out all the way around. Now, this is going to be our capture device for capturing some gases. And what we're going to do is we're going to put the balloon over the mouth of the bottle. Now, this is kind of tricky. You may need an adult to help you hold the bottle, or you might want to put it between your knees if you're sitting by yourself, just some way to secure the bottle so that when you put the balloon over the mouth, it doesn't spill. All right, it's a little tricky. And for me, I really like to kind of stretch the mouth of the balloon with my fingers like this first, and then put it all the way over. All right, all the way over, and try to pull it down as far as you can. And if you can't get it all the way down past all the rings, that's okay, but do what you can to try to get it down as far as you can. You might even be able to roll it just a little bit, okay? So we're going to take the balloon, we're going to put it over the top of the bottle. We have our yeast and our sugar and our water, and we've just set up our experiment. All right. Now we're going to talk about why we did all these things in just a few minutes, but this is just the start. And we're going to set this up so that we can come back to it and see what happened. Now remember that yeast is actually a type of fungus, and that's why we're using it, as well as the mushroom fungus that we gave you here. They're just two parts of the same family. They just look a little bit different. So our experiment with our yeast, our sugar, and our water, and our capture device up here. All right. If you are ready to go on, then tell me in the chat, what is your favorite animal? All right, I'm going to give you just a minute for everybody to catch up. If you're ready to go on, tell me what your favorite animal is in the chat. Now make sure that you put this bottle where it's not going to leak and you can get it, the surface wet, away from your electronics. Oh, I see some great animals coming in with cheetahs and birds and foxes and turtles. Great job, everybody. Thank you so much. That's awesome. All right. So you've got something else in your kit that we're going to do in just a minute. And that is, you're going to need to find your little lens and your putty. Now, not everybody's going to use the little lens and the putty. You're going to use it if you have one lens on your camera. And to do that, essentially, we're going to use this as, think of it as a magnifying glass for your phone. Now, some phones have many different camera lenses, and this won't work at all. And if you don't have a phone at all, that's okay. You can just use your eyeballs. We just gave this to you as a way to make a magnifying glass out of your phone if you want. But you really have to have a phone with just one lens. All right, you may have one flash and one lens. If you do, and if you want to try this, what you can do is take this little piece of putty, remove the piece of paper, and you're going to take the putty and roll it in your hands, and you're going to make a little snake out of it. Now, when you make a snake out of it, you're going to roll enough so that you can put it around the lens of your phone, like a little donut. I'm going to show you here. You're going to make a little donut. And then you're going to press it onto your phone. Now, remember to get permission from your parents before you do this, but you can try this later if you find that we're going too fast, and you can try it with other materials. But what you're going to do is after you put that little donut over your lens, then you can push the lens we gave you into it, sort of like you're putting it in the center of the donut. All right. And that's going to be setting on your cell phone camera. Now, you don't have to use this. Remember, if you don't have a cell phone or if you don't have permission to use it, it's just another fun tool we're giving you, like a magnifying glass. So you can use your eyes to look at the gills of the mushrooms in just a minute. You can just use your cell phone and the pictures and camera, or if you want to, you can try putting this little lens over the camera on your phone. All right, just if you want to try that, but not required. All right. Okay, so we have our experiment running. We have our gills set to the side. We have our little lens ready to go. I think we are ready to meet the scientists. All right, let's go to the field and meet Melissa and Eranga, our scientists, while our experiments are running.
Hi, I'm really excited to be here with you today. My name is Dr. Melissa McCormick, and I run the Molecular Ecology Lab here at SERC. Now, molecular ecology just means that we use DNA to study a lot of different processes in ecology. And I use DNA especially for studying fungi. And the reason I do that is because a lot of fungi are very, very hard to see with our naked eye or with even a microscope, and it's very hard to identify them. So that's what I use the DNA to do.
Hi everyone, I'm Dr. Eranga Wettewa, and I'm also very interested in learning about fungi. Specifically, I'm very curious to know how these plants find their fungal friends and also their enemies. So I'm working together with Melissa as a research fellow in her Molecular Ecology Lab, and today you can learn some very cool stuff with us. So come on, let's go for a walk.
Today I'm recording this from one of my field sites, and I wanted to start out here in the woods to show you some of the places that I find fungi. But later we're going to go back to the lab and I will be joining you live to discuss some of the fungi and some of the things that we study about fungi. So when I'm out here in the woods, I see a lot of the different but really important things that fungi do. And they do a lot of really important things. And one of the important things that they do is to feed plants. And for some plants, the fungi are so important that the plants can't grow there unless the right fungi are there.
Hey Melissa, what kind of plant is this?
This is a great example of a plant that needs fungi to grow. This is an orchid called a rattlesnake plantain. And the reason it's called a rattlesnake plantain is because when it sets fruit, the seed capsules look like the tail of a rattlesnake. And orchids, all orchids, need fungi, and particular fungi, in order to grow. And those fungi are feeding on the leaves and the decomposing wood, and they're feeding it to the plant. And that's how the plants are able to grow. And all orchids, because they need specific fungi, their presence or where they grow is really driven by where the right fungi are. So in nature, every orchid depends on particular fungi to feed it, and many other plants do too. Those plants can't grow without their fungal helpers. This rattlesnake plantain can only be fed by one type of fungus, and then it transfers the nutrients, things like vitamins and minerals, to the orchid by decomposing the wood and leaves. And fungi can even communicate with each other and the plants that they live with. The fungi can send other fungi and plants signals through the soil, saying if it's dry, or if something is eating the plant, or it's under attack from something else. Why don't we take some of the soil and decomposing wood back to the lab and see if we can find fungi?
Oh, look at all the tree roots in this soil. All the trees need fungi to get nutrients too. And this decomposing wood has lots of nutrients in it. The fungi help them get nutrients and water, and can even connect multiple trees together, letting them share nutrients.
Follow us back to the lab where we'll set up your experiment.
You can look at mushrooms up close with me.
So what you're seeing is Melissa and Eranga were walking in the woods right here at SERC, and they were showing you some fungus and orchids that are out even right now in winter. And so we have orchids, yes, right here in Maryland, but they don't look like the kind that you get from the grocery store. The one that she was pointing out is an evergreen orchid, and it relies on fungus. That's part of what they study in the lab. And so what we're going to do is we're going to learn about how fungus helps trees and plants. And we're going to see the fruit of some fungus in just a moment with Miss Eranga, and she's going to help us with the first part of our experiment with dissecting the gills of the mushroom. All right, so go ahead and get your bag with your mushroom ready. Now, Miss Eranga is going to explain how this is related to the types of fungus that are found in the forest. With that, I'm going to turn it over to her for our mushroom dissection. All right.
Okay, hi again. So I'm Eranga. So hope you all are very excited for an investigation up close and personal with the mushroom today. So before we begin though, you'll need to make sure you have your Science to Go kit open and ready to use and a cell phone with a camera. Before we begin, make sure you get permission from your parents to use the phone, or you can ask for them to help you. Okay, now let me share my screen. So I have my rehydrated or moistened mushrooms ready to go with some more little items that you also have, like the forceps, some napkins, and an index sheet. So I guess you can take your little baggie with the mushroom and water and see how they look now. And let's go ahead and take the mushroom pieces. You can use your fingers to take one of the pieces out.
And if your fingers are too short, you can also use your forceps or your tweezers to get into the bag to take your mushroom pieces out.
Yeah, that's right. And also, now, can you remind me what a mushroom is again? Are they some kind of fungi? What do you think? Yeah, mushrooms are like the flowers of a plant, but instead of making pollen like a flower does, they make something called spores. So spores are dust-like and they fall to the ground where they will make new fungus. All right, so you can take your hand out, which is also included in the kit. And there's a diagram of a mushroom. Like this. And can you try to identify the parts of this mushroom? I also have a real mushroom here. So you can see the different parts of this mushroom, the cap of the mushroom, the gills of the mushroom, and this is the stem, right? So you try to identify the different parts of the mushroom. So today, what we are looking at is a piece of this gill. That is what we are going to look at. All right, now back to my
Now, while Eranga gets back to her camera, remember that the mushroom pieces that you have may look different. Everyone's a little bit different. So the things she's sharing with you may not look exactly alike, and that's okay. You may be looking at different parts of the gills. So when she shares these images with you, just remember that they may look a little bit. And Miss Eranga, would you like them to put their mushroom piece on the napkin or the note card?
Yeah, you can keep your piece of gill on the index sheet or whatever the paper you have with you, like this. Is it clear now? All right, and now you can take your little piece of napkin and try to damp it and just pat it gently on the mushroom, just like this. And can you see what happened? So you'll see a big brown color patch on your napkin. Look closer into that. Pat it on the piece of gill gently and see. I'm going to focus a bit more onto this big brown patch.
Can anybody guess or make a hypothesis about what we're looking at? If you think you know the answer, you can put it in the chat.
Yeah, what are these? What do you think these are? And also you can also observe the color change in your little baggie. See how it changed from just colorless to brown color? I guess that same thing happened in your baggies too. All right, now we are going to use that little phone camera or the microscope that you just made with the help of Karen. So you can use that little camera to focus on this little brown spot.
And remember, if you don't have a camera, you can use just your phone's camera, just normal, if you didn't use the glass lens, or just use your eyeballs. Just look really close in good light.
Yeah. If you're using the camera, move the camera closer and further from the paper towel until you can see things. You can also try zooming in and out a bit using the cell phone camera, just like you focus on something when you're taking a photograph from the camera. Can you all see these brown, small dark patches or circles? So these are the spores that came off the mushroom gill. They just came out from this gill.
Oh man, we had a lot of great guesses. Many of the people guessed spores. So good job, everyone. If you guessed spores, pat yourself on the back.
Yeah, that's great. All right. And until you experiment on that a little bit, I'm going to show you another great picture of actual spores. And this is how you see it when you look through a microscope, actual microscope. So can you see these little oval shaped things? These are the spores, and they are very, very magnified. So you won't be able to see this kind of spores from your phone camera, but this is how you can actually see them when you look through a microscope. All right, back to the spores. And now we're trying to remove a little piece of the gill using the forceps or the tweezers. So you might have something like this. I have something like this. So I'm going to use this little forceps to remove a piece of this gill. So it's better if you can try to take off a single piece of gill, but it's okay if you can't, or if you take off a part of the gill or maybe more than one gills, that's fine. So try to take off a little piece from the gill, and you can place it on your index card, just like that.
Now, if you need help finding the gill, you want to look for something that looks a little lighter. That's the cap of the mushroom. And then hanging down underneath the cap of the mushroom should be very, very fine slivers. And they look almost like the gills of a fish, but these are the gills of a mushroom. And they're very long and kind of thin and like the teeth of a comb almost, but very, very close together. If your mushroom doesn't look quite like this, look for a pattern. You might see kind of a bumpy pattern, or you might see the comb pattern. Do the best you can and try to find the gill. But if you can't find it, that's okay. You can just watch Eranga for right now.
Yeah, and also, I think you all love mushrooms in your pizza. So next time when your parents bring mushroom home, so try to look at these mushrooms. So this is what are called gills. dark brown part. So that is what we are looking at now. So try to take a little piece of that, and you can keep one of those little pieces on the index shade, just like that. And now again, you can use your cell phone camera. Just make some observations of the gill. If you're having any problems or difficulties, you can type your questions into the chat. So what do you notice about the gill? Yeah, you may also see spores attached to the gills if you look very closer and if you can focus them very well. All right, and if you have multiple pieces of gills, it's okay. You can try to compare each of these parts and see if you see any difference.
And Miss Eranga, we have some great observation. Some people are saying that they see soft, it's kind of soft. Some people are saying that they're seeing little dots or spores coming off of it. So we've got some great observations.
Yeah. So yeah, if anybody is having trouble following our own steps, like if you're not catching up with us, it's okay. Just carefully listen to us and observe what we are doing. You can try and play with it later sometime. So don't worry about that. Just look at what we are doing carefully. Okay, I think now it's time to remove the mounting putty and lens from your cell phone. I think you all have done a great job.
Sorry, just to wrap up, Miss Eranga, when they're looking at the gills, you say the spores are on there. Are they attached to the gills? Do they fall off the gills? What happens? Because I saw stuff in the water, right?
Yeah.
How did those get there? What happened?
Yeah, so there inside these gills, a lot of spores, like millions of spores are inside these gills. So just like a flower is producing a lot of pollen, these are producing a lot of spores inside these gills. So when we put them in the water, they can come out to the water and like disperse in water. And that's why, that is one reason for this color change. There can be other things like pigments, but spores are one major thing that cause this changing of color. So yeah, they're coming out of these gills. Yeah, so since that, you might be able to see some spores when you are looking carefully through at these gills. Yeah, and there's one more thing we can do with this same things, Miss Karen. Do we have time to tell that?
Very quickly, because we have to get over to Miss Melissa.
Yeah, okay. So since we are running out of time, I'm just telling you briefly. So in your handout, you have instructions to do this step. So you can use the same gill piece that you just used, and you can use that to grow your own mushrooms at home. So all the instructions are in that handout. So you can try by yourselves with the help of your parents. So yeah, just try that at home. And now I think it's back over to Karen. And yeah, let's see the results of your yeast balloon activity.
Actually, thank you so much. That was really neat. So Ms. Eranga just introduced us to this idea that mushrooms are like the fruit of fungus. Now, mushrooms aren't out year-round. Usually they come out around the fall. There are things called mushroom farms where they grow them, and you can even grow them in your own refrigerator. Now, I want to know more about the types of fungus, though, that our scientists like Eranga and Miss Melissa are studying. So I think before we check our balloon, because, oh, I see it doing some cool stuff. Before we check that, let's talk to Miss Melissa and see if she can tell us a little bit more about the stuff that the mushrooms grow on. Miss Melissa, can you tell us more?
But I wanted to tell you a little bit about sort of what I do with fungi. So fungi come in a lot of different sizes and they do a lot of different things. And sometimes fungi are so small you can't see them, like the yeast that you worked with in your experiment. So if you were to try and find the individual yeast in there, they'd be very, very small and you'd need a microscope to see them. But some other times they're bigger, like the mushroom you dissected, or like some of the ones that you eat, like we have here. So I have a shiitake mushroom and an oyster mushroom, and the sorts of mushrooms that you might have on your pizza. But I was also going to show you, and for some reason, let's see here. So some of the fungi that we grow in the lab. And so these are, as Ms. Karen told you, the mushrooms that we see are sort of like the fruit, and underground, there's a whole lot more of the fungus. And these are some of the fungal hyphae, or the strands of fungus that would normally be living in the soil. But this is what we grow in the lab and what you might be able to grow if you grow some fungi from your spores. But the other thing is that sometimes fungi are a lot bigger. So for example, type in the chat what you think the biggest living creature is. Let's see, somebody said a whale. Yeah, I got a lot of people who are saying whale. So the biggest living creature is actually a fungus. It's not a whale. In Oregon, there's a honey mushroom fungus where the single fungus growing in the soil, the hyphae of it that you can see here in the soil, are almost 2.5 miles across. And scientists think it's over 8,000 years old. Now, if you wanted to walk 2.5 miles, it would take you over an hour. That's how big this fungus is. Now, I study what different fungi do to help plants grow. I try to understand how fungi feed plants and how the fungi that grow in an area can determine what plants grow there. So the orchids that we saw in the field earlier in the program are plants that need particular fungi in order to grow. So some plants need particular fungi, like the orchids we saw. Other plants need a couple of different fungi, but almost all plants need at least some fungi in order to grow. Now, Eranga is working to understand how orchids and fungi work together, how they communicate with each other to grow together. And I want to know how fungi and plants work together to allow plants to grow in different places. I also want to know how we can grow more fungi in order to help grow more native orchids. Because a lot of orchids are very, very rare. They're very hard to find. And to be sure that they can survive or grow more to be able to put back into the wild, we need to be able to be sure to save the fungi that they need too. Now, the fungi feed plants by attaching to their roots underground, and they form a big network, kind of like the internet, where they can talk and communicate. So when you see it, like for example, when you see a tree, you only see what's above ground, but there's more tree below ground than there is above ground. And all of those roots below ground are connected with fungi. And they can send signals. Trees can send signals to other trees, and to other plants can send signals to other plants, basically either asking other plants for nutrients or letting another plant know that they're under attack by insects. So they should start up their defenses. And they do this through their fungi because the roots themselves are not all the way together. Now, when I learned that fungi could even allow plants to talk to each other and to grow in places that they couldn't grow alone, I realized how much of learning why plants grow where they do depended on solving the puzzles of why particular fungi grow where they do. And that's something that we really don't know very much about. I also want to know why only some fungi feed particular plants. Why can't all fungi do it? Or why do any fungi do it? And I love puzzles. So how many of you like puzzles or mysteries? You can type that in the chat. How many of you like that? I see at least a few people do. Now see, I love puzzles. And to me, science is sort of like the ultimate puzzle. And it's a puzzle that I, in some ways, get to design myself because I come up with the questions I come up with the puzzles that I want to try to solve. And so understanding how all of these different creatures work together, and in this case, I focus mostly on plants and fungi, but understanding how they work together is sort of like the ultimate puzzle to me. And that's what I work really, really hard to understand. And so for me, the plants that I mostly focus on are orchids. And well, orchids are beautiful and a lot of people have seen them, but a lot of people see them in the grocery stores and they don't so much see them out in the woods. We do have lots of orchids in North America that are native here. They grow in the woods. And We also have some examples. I don't know if anybody has seen them already, but we have some paper models of orchids called orchidgami that we make paper punch outs that you can punch out and make them into orchids yourself. And they have some information about the orchids. And what we're working to understand is we're working to teach people about the different plants that we have, but also about the fungi that they need in order to grow, because they're all sort of interconnected and they all make this ecosystem that works. Sorry, there's somebody who spelled fungus wrong in the chat and apologized for it, because we all spell fungus wrong every now and then. But so one of the things that you can do if you're able to grow fungi from your spores is another thing that you can do is you can actually make a spore print if you have a mushroom cap that you've gotten from the store. So this is the top of a shiitake mushroom, and you can see the gills in the bottom of it. And if you were to take this cap and put it on top of a white piece of paper, or I use some aluminum foil here, you can set it down like that and put a damp paper towel over it and put it in a plastic baggie. You can actually get it to shed its spores. It will release them. And this is very hard to see on a camera, but you can actually get a print that shows the spores or the mushroom where all the gills were. So you might want to try that at some point if you have a chance. And so with that, I think I will turn it back over to Karen. And I think we're going to go see how your experiment is doing.
Oh man, so many good facts about fungus. We've learned about their fruits. We've learned about the stuff that's underground. Let's find out about what our type of fungus was just doing. So I want everybody to take a look at their bottles. Do you notice that something happened? With mine started to blow up the balloon. Tell me what happened to your bottles in the chat. What do you notice? When you look at the foam in here, is the bottle more full than when we started? What's going on? Does anybody know? Write your answer in the chat. What do you think caused this bottle to fill up? Was there just magic? There was science magic in there. What do you think is in the balloon? Don't squeeze it too hard. What do you think's in that balloon? Well, yeast are kind of like us. They are living things. When they're dried and they're put into packets like this, they essentially go to sleep. And just like us, when we wake up in the morning, we like to eat breakfast and have some sugar and food. We need to drink some water. We need our oxygen to breathe, and so do yeast. And that's what we did. We woke our yeast up. And so when we put the yeast in the warm water here in our bottle, and we gave it sugar, we gave it food, we woke it up, and all of a sudden it started to breathe. And yeast breathe sort of like we do. They use oxygen and they give off carbon dioxide, just like us. So if you look inside the bottle here, you're going to see bubbles. And those bubbles are carbon dioxide that are being given off by the yeast. Now, have you ever cooked with bread before? Cooked bread or used yeast before? Well, that yeast is what we use to make bread rise. It's essentially what gives the bread lift. So if you cut a piece of bread and you look inside, you may see holes, right? This is a picture of some of the holes you might see. And that is because when you mix flour and water and sugar all together with the yeast, it makes the yeast wake up and start releasing these bubbles. When you bake the bread in the oven, the bubbles are still there, but the yeast are baked out, so they're no longer alive. All right. Now, here's my question for you. If you were to leave this just sitting around, what do you think would happen? Any ideas? Well, I'm not going to tell you because science is all about finding out things. So if you would like, you can leave this sitting in your sink. Make sure it's somewhere that if it drips or gets gross, it's not going to get you in trouble. But let's find out what happens if you leave that sitting out. And you can share that with us on social media or share that with the people in your family. Find out what's going to happen. All right. Now, the cool thing about this yeast that's in bread, if you didn't have yeast in bread, it would be flat. That's the kind of crazy thing, just like a cracker. And so we need yeast to make bread rise and to be light and fluffy. All right. Now, so I'm going to let you all figure out the rest of the experiment. But if you are going to clean this up, make sure that you hold the bottle over the sink. And what I want you to do is when you take that balloon off, I want you to smell and see if you can smell actually what smells like bread. It's the yeast giving off the carbon dioxide. You're going to smell that smell. It's very distinctive. All right. I think mushrooms and yeast both have very distinctive smell. All right. So we've learned about today, just to recap, we've learned about mushrooms being the fruits of fungus. We've learned that yeast are a type of fungus too. They're just single-celled and we use them in baking and in food. We've learned that fungus is in plant roots and there's more plant below the ground than there is above ground, right? And that fungus helps the trees and the plants communicate. Well, I've got a challenge for you. I want you to see if you can go outside next time you're out and it's not raining and see if you can find fungus by rolling over a log or opening a piece of bark. Look with your eyes and see if you can see the white strands of fungus in the soil. All right, so go on a soil scavenger hunt for fungus. But remember, with mushrooms that are out in the wild, don't just pick them or touch them without permission. You only want to touch or eat the ones that are in the grocery stores and that are safe, that your parents say are safe. So just to recap, fungus are our friends. They're found in food, like bread. They're actually used to help make some cheeses. They're used in medicines, even like antibiotics, which I think is super cool. It makes us feel better. And they're even, get this is gross, found between our toes. Ugh, yes. And sometimes if you let that bread or that fruit sit too long in your refrigerator, you're going to find fungus. Fungus is all around us in the air and spores that we can't see, but they're helpful. They help our forests and they help us. So fungus are our friends. All right. So with that, I want to say thank you for joining us with our experiment. I really can't wait to hear what happened to your fungus. I'm sorry, to your yeast. And I hope you will go outside and do a fungus scavenger hunt. And with that, we had some amazing questions in the chat, and I really would love for Melissa to answer some of those for us. So I'm going to hand it over to Joanna to ask those questions and Melissa.
And if you have more questions and want to share them in the chat or Q&A, please feel free. We'll have a little bit of time to ask some of them. So Melissa, I guess my first question for you that came up in the Q&A is, can fungus grow in any climate?
Yes, so fungus grows pretty much everywhere. There are different fungi that grow in different places. So just like, I mean, there's yeast everywhere, different kinds of yeast that might make good bread, it might not, but there's different kinds of fungi everywhere. So different mushrooms will grow in different places. So just like with plants, if you have a plant that can grow in, say, Maryland, it might not be able to also grow in Texas, but there will be a different plant that can grow there. And so fungi are the same way.
Awesome. Okay, up next. So I know you were talking about fungus and Maryland orchids. So what fungi can grow with Maryland orchids?
So not so much the ones you eat generally. There are a few orchids that can use the fungi that are ones that are sometimes eaten and that make mushrooms, but most of the fungi that support our Maryland orchids are ones that grow in the soil and they don't actually ever make a mushroom. So they might look more like this one here, where if you were to see it up close, you would see that it's just thin strands of fungus, the fungal hyphae that would be growing in the soil or on decomposing wood. And that's mostly what feeds our orchids.
So kind of related to that, somebody was wondering, what does fungus underground look like?
Well, it looks a lot like some of the things that I have on these plates here. So it looks like little thin strands, and sometimes it's got bright colors. And sometimes, a lot of the time, it's kind of a pale cream color or white, but they're very, very thin strands. They can be very hard to see. So if we had an amount of soil that was about the size of, a lima bean, that might have 150 different species of fungus in it, different kinds of fungus. And you wouldn't be able to just look at it and tell which was which. And so that's why I use DNA to identify the fungi, because I can pull out the DNA sequence that goes with each of those fungi and figure out which different types of fungi are there.
That's really cool. So somebody wanted to know if fungus is a type of plant or dirt.
Neither. So fungus is its own type of creature, and it's actually more closely related to animals than it is to plants, even though we think about it as being more like plants. There are some fungi that live in the ocean that actually swim, which is kind of surprising. And there are some fungi that move fast enough that you can almost see them move yourself just with your naked eye. But most of what we see is it grows mostly in the soil. It can make mushrooms for fruits, and it's its own kingdom. So it's not really very closely related to either plants or animals, but it is a type of creature or type of organism.
All right. So somebody wanted to know, can fungus grow in like the Arctic?
It can. In fact, there's a lot of fungi that grow in the Arctic. And one of the things that they're very concerned about with warming climates and the warmer summers that they're having there and not such cold winters is that the fungi wake up a lot. And the fungi, a lot of what they do is they decompose or break down the dead leaves and other tissues that are on the soil or from the organisms that grew there and died. And so when they break that down, now that carbon is not stored anymore. It's actually released to the air. And then it makes it even warmer the next year a little bit. And so that's one of the things that fungi do is they take dead tissues and they turn it into food that other organisms can eat. But in doing that, just like our yeast in our experiment produced CO2, carbon dioxide, as a part of their respiration, other fungi are doing that when they break things down too, and some of the CO2 goes back into the atmosphere.
So a couple people had some questions about the yeast, so I figured I can ask them now. So two of them that came up quite a bit is yeast a living thing? And then why does it smell when we did the balloon experiment?
So yeast is a living thing. And what you smell is sort of like as a person, when you breathe out carbon dioxide. Well, the yeast is breathing out carbon dioxide too. And just like sometimes your breath may not smell so good, well, sometimes what the yeast breathes out doesn't smell so good either. And so that's sort of what we're smelling.
So another question somebody had is, do fungi and mushrooms need light to grow?
They do not need light to grow because they can't, just like plants use light for food. Fungi can't do that. It takes plants and it takes the green chlorophyll that they have in order to do that. And the fungi cannot do that. So a lot of fungi actually grow best where it's really dark.
So someone else wanted to know, and I think this might be kind of a big question, how many different fungi are there?
So they think there's about 1.5 million different species of fungus. So there are more different species of fungus than there are all plants, animals, and all the plants and animals put together.
Wow.
There's amazing. A lot of them. Yeah, there's a lot of them. And so a lot of people are really surprised by that. And a lot of the fungi have not been described yet. We don't really even know. I said they think there's 1.5 million, but that's a guess because so many of the fungi live like down in the soil or down in the bottom of the ocean or in places that we can't see them. And if we haven't seen them, then we don't have a species name that we can stick on it. I mean, the fungi that I work with that feed orchids, every fungus that we get is a species that has never been described. There is no name that I can put on it. If somebody asks me what fungus feeds that orchid, I'm gonna say, well, it belongs to this genus, but there's no common name, there's no species name to put on it. And so it can be very hard to figure out just how many species there are.
So I think we have time for maybe one or two more questions. So one person wanted to know, how do fungi communicate?
Well, they communicate in that they're sort of almost like a tube. The fungal hyphae, those strands, are just sort of like a tube or almost like a long nerve fiber. So things signals that starts on one end of it can sort of propagate and almost diffuse to the other end of it. And so it's just like it's just like communicating with chemicals or with smells. You know, if you wanted to say, oh, well, when you smell lemon, it means you need to do this. Well, if somebody opens up a lemon on one side of the room, eventually, even if you're on the other side of the room, you'll smell it and you'll go, oh, that means I need to do this.
All right, so I think the last question we're going to have time for today, are any fungus extinct?
There are some fungi that are thought to be extinct, but as I said, it's hard to tell. So with fungi, it's easiest to tell if you see the mushrooms that the fungus is there. But if the fungus was never described, you don't know if it disappears. And if it's just not producing mushrooms and you're used to seeing mushrooms from it, then you may not see that it's still alive and is hiding out somewhere. So it's very difficult to tell, but there are some species that they think are extinct.
That's really cool. All right, and I think that is all the time we have for questions today. Thank you, Karen and Melissa and everybody who helped put this together. It has been so much fun and so exciting to see everyone reacting and seeing their balloons blowing up and the mushroom dissections and everything else. For more programs from the library, you can check out our website and we'll put a link in the chat. And we'll also be sharing the recording of the video that we unfortunately weren't able to make work during the program afterwards. And eventually we'll also be sharing the recorded video as well. And thank you to all of our participants for coming and joining us today. I hope you had fun.
Thank you all for joining us.
Bye. Thank you for joining us.